Hi everyone, this is Xiaotong Lin. In this video, I'll be talking about my project on probing toxic protein aggregates in Alzheimer's disease using an immunoquantitative polymerase chain reaction approach. This project is supervised by both Dr. Francesco Prillet and Professor Ramon Villa. And I would like to start with a short introduction of Alzheimer's disease and why probing these aggregates as biomarkers is important. Alzheimer's disease, also called AD, is a neurodegenerative disease, and it is the most prevalent form of dementia, where dementia is currently affecting more than 50 million people worldwide. Even more, this disease is expected to cost the world around $1 trillion in 2050, which, if stuck together, could go 1/4 of the way to the moon. One key hallmark of AD is the formation of amyloid beta synapse plaques observed in patients' brains. These plaques are formed from aggregation of A-beta monomers, where different species are formed during the aggregation, including oligomers, protofibrils, mature fibrils, and eventually the synapse plaques we have seen earlier. Studies have shown that A-beta oligomers, a transient species formed during the aggregation, have been identified as the most cytotoxic intermediates formed during the misfolding of A-beta. The key underlying mechanism for the formation of this oligomer species is still unclear, where this information could provide insight into the pathology of AD and potentially for a therapeutic approach towards AD. Current methods in the field are powerful tools for probing various aspects of A-beta aggregation. However, many of them, including PET analyzer, are limited by their high expense and the requirement of a trained personnel for the operation. Therefore, a detection and quantification method that is high throughput and oligomerase physics could help to aid the study of oligomers and could potentially be adding to diagnostic tools. This understanding of oligomerase species is hindered by their sparse, transient, small, and heterogeneous nature, and the fact that they are spread throughout the brain, making probing these oligomerase species really difficult. At the White City campus, I work with the acrylic group and the violet group, where the acrylic group is specialized in using antibody-based technology to understand misfolding protein mechanism, and the violet group is specialized in developing novel tools to interrogate biological systems using medicinal inorganic chemistry, chemical biology, molecular recognition, and molecular imaging. In my project, I'm using an iqPCR approach to probe the A-beta oligomers. In this setup, a probing antibody is used to detect the tyrosine oligomer species, where the signaling antibody is attached to another probe that gives out a fluorescent readout in real time. The PCR is checked using this fluorescent readout, and a graph like the one on the right-hand side is generated. And when there is high quantity of oligomer species present, the PCR cycle number will be found earlier comparing to the other samples containing lower number of oligomer species. This project works towards a quantitative detection method for the A-beta oligomers, which are difficult to track. It also has the potential to be expanded to a wider scope of misfolding protein. This method will hopefully aid the field with a high throughput method for detection and quantification of A-beta oligomers, and moreover in the future, the misfolding protein. Thank you very much for listening.